Okay, so this event is being recorded and it will be made available afterwards. So you'll get an email in the coming weeks with a link to be able to watch it back again. It'll also help those people who, for whatever reason, weren't able to join us today. So, so I can see people are still logging in. Whilst that's happening, I'll just quickly tell you how we're going to run things in Zoom today. So as a couple of you very kindly used the chat box to let me know you can hear me. If you have any technical problems, if you can't hear us or there's any kind of issue, put it in the chat box. Myself, Qasim and Rana will be able to see that. So let us have a message. Please put your questions in the Q&A box. If you look at your controls, you'll see you've got a series of controls. There's a Q&A box. It's really great if you put your questions in there because we can see them as they come through. We might want to gather them up together, or you might ask a question which one of the panelists will actually answer later on anyway. So it's really helpful for us if you put your questions in the Q&A box. You can ask your questions anonymously. And you can ask your questions at any point in the session. What I'm going to do is I'm going to wait for people to log in. It normally takes a minute or so longer. And then I'm going to hand over to my colleagues, Dr. Qasim Rafiq and Dr. Rana Khalife. And Qasim will start off with his presentation. Then Rana will give her presentation. And I'd just like to say, normally in these sessions, we don't get to answer all the questions, but Rana and Qasim will be available to talk specifically as in their roles on the MSc stem cell and gene, the commercialization and manufacturing and commercialization of stem cell and gene therapies. So they will be available on Friday. So I'll put a link in the chat. So now that it looks like we've got most people logged in who wants to log in, I'm going to hand over to Qasim. If my camera goes off at any point, it's to save bandwidth. I will be available in the background. Okay, Qasim, over to you. Just want to turn your audio on, Qasim.
Qasim, you're muted.
I am muted. I should have realized that. Well, thank you very much, Kim, and thank you all for attending today's lecture. It's a pleasure to have you all. And as part of this series, what we're trying to do is give you an overview of some of the content that we would cover. So it's going to be very introductory, but some of the content that we would cover to either undergraduate or postgraduate programs. And as Kim mentioned, I'm the program director for a new master's program which started in September 2020 and has been a major success despite launching under COVID, focused on the manufacture and commercialization of stem cell and gene therapies. And we're going to cover some of that in a bit of detail today. So before I begin, I'd like to introduce myself. So I'm an associate professor in the Department of Biochemical Engineering at UCL with a research focus on cell and gene therapy bioprocess engineering. And I'm also program director of the new master's program that I alluded to earlier. I'll hand over to Rana, who can introduce herself.
So hi everyone. So my name is Rana Khalife. I'm the regenerative medicine minor coordinator, and I'm leading some of the courses undergraduate and postgraduate with the MSc.
Fantastic. Thank you very much, Rana. So Rana and I will be co-delivering this lecture. So I'll be covering the first half focused on advanced therapies and gene therapies. And then Rana will discuss a bit more detail around tissue engineering, regenerative medicine, and stem cells. So before we do that, I'm going to briefly introduce biochemical engineering and kind of why is it important and how does it relate to stem cells, gene therapies, advanced therapies, regenerative medicine. So really, biochemical engineering for me is the critical discipline which allows us to make discoveries a reality. So that's why it's kind of highlighted there in red. So the ability to really transform and translate what we discover in the lab into a practical reality that can benefit patients, that can change the global, or address some of the global challenges that we're facing, on a large scale. And it's not just healthcare. Obviously, we'll focus extensively in this lecture on healthcare applications, but you think of the challenges we now face as a global society. And for me, biochemical engineers are very much at the heart of it. I'm not just saying that because I'm in the department or that I myself 15 years ago graduated as a biochemical engineer from the department. But from my perspective, this is probably one of the most fundamental disciplines moving forward when you look at the state of the global challenges we're now facing. I think first and foremost, it goes without saying the role that biochemical engineers have played in addressing and developing COVID vaccines on a transformative scale. We went from the point where in January 2020, we had no vaccines being manufactured for COVID, and we kind of come 12, 18 months later, and we now have a multiplicity of vaccines now currently available, which people are receiving. And I've got my first jab booked this Saturday, so I'm very excited to have that. But for me, biochemical engineers have been fundamental in being able to get to that point where we're not just producing, a handful of vaccines, or we're producing enough to treat 100, 1,000, or 1,000,000 patients. We are looking at having to produce billions of doses to treat the global population, and it's biochemical engineers who are making that a reality. Likewise, when you look at the manufacture of other therapeutics, and we'll go right the way through to what I would say was kind of the first hugely successful and huge manufacturing challenge of producing antibiotics, so penicillin. We all recognize that Sir Alexander Fleming is credited with the discovery of penicillin, which he almost certainly did discover. But to turn that into a reality required the role of chemical or biochemical engineers in order to take this discovery of penicillin and be able to mass produce for a global society. And that's kind of the role of biochemical engineers. I think we're very much at this stage with cell and gene therapies where we've shown some of the clinical and scientific promise. These are transformative therapies. We're curing things we've never been able to cure before. But it requires biochemical engineers now to turn these discoveries into a true commercial reality, and something that patients can actually access at a cost that is affordable. And so when you look at the things that biochemical engineers are often working and manufacturing, if it's in the healthcare or medicine space, it's vaccines, antibiotics, therapeutic proteins, regenerative medicines, and cell therapies, which will obviously be a focus of today's lecture. But also when you look at the other global challenges we're facing as a society, there's one major challenge around the development of sustainable fuel sources, so things like biofuels and so on. But also another key challenge we're facing on a global scale are things like climate change, but also the how to deal with all of the plastic waste that we're now facing. And in all of these areas, they're all using biochemical engineering or biological approaches, which requires the biochemical engineers to lead much of this work. And that's why when you look at existing large, petrochemical companies, BP, Esso, Shell, they have never recruited more biochemical engineers than they have at this moment, because they can see what we can all see, which is that the world is changing, the global challenges are changing, and we need engineers who understand the fundamental biology to be able to address some of these challenges and issues. Hence why we're focused on this within the biochemical engineering department, particularly for cell and gene therapies and for stem cells. So why, now moving more specifically onto the stem cell and gene therapy activity, and more broadly the field being known as cell and gene therapy, why cell and gene therapy manufacturing? Well, really it comes down to kind of two key aspects. Firstly, this is a clinically pulled high value manufacturing activities. What does that mean? That means firstly, it has significant clinical potential. So you'll see in some of the videos and the discussion later today that much of what we're doing is manufacturing therapeutics, which will have significant clinical impact and treat things we've never been able to treat before, patients who have failed every other treatment option and are now making fantastic recoveries from their conditions. But also it's a manufacturing activity, so it provides significant investment back into the economy. Manufacturing is one of those sectors where it has a big impact on society, in that you create more jobs, it creates products which obviously can be sold and commercialized, which provides a significant amount of investment. And as a result, the UK has made cell and gene therapy a key priority, cell and gene therapy manufacture, I should specifically say, a key priority for the economy moving forward. But also there's a big focus on how can we address some of the dated and stagnant technology platforms, move towards automated processes, move towards more data science-based approaches, and establish process control systems to address the current poor control systems that we have at the moment, and kind of move to a more something that is very manual to something that, an industry that is highly automated, very precise, and has a big impact on our lives. So I just want to kind of give some of the highlights of cell and gene therapies and just kind of how things are transforming. So this was an article back in 2018 where BBC News covered some of the work that had been undertaken at UCL, where the NHS agreed a deal with one of the first CAR T companies to offer CAR T therapies on the market, and we'll come to what CAR T therapies are in just a second. And this is an article a year later where one of the patients who was suffering from a terminal condition was offered a CAR T therapy and is now making a full recovery. And you'll see many examples of this throughout. So the other key thing I wanted to highlight is this is an industry where there was a lot of clinical excitement, a lot of scientific promise, but we weren't seeing products get regulated or come onto the market. But that is now changing. So this is taken directly from the US FDA website. So the FDA is the Food and Drug Administration, the US regulator for healthcare medicines. And you can start to see a number of products now emerging, which have been approved by the US regulator as for cell and gene therapy products, which have been approved for use in humans. And this is then, very quickly followed up by the European Medicines Agency, which is the European regulator, the EMA, which have also approved a number of gene therapies. And just to kind of highlight again, so this was in Reuters, that deal I mentioned earlier, so Novartis being able to offer their CAR T therapy to UK patients via the NHS. So before we go into detail, I do want to address one of the issues we have in the sector, which is the names for these therapies. So there is a wide range of nomenclature and a number of different terms used to describe what we mean by these therapies. I'm just going to very quickly just give a run through an overview. The broadest term is advanced therapies. So these are therapies which I think many recognize are now far more advanced than things we've used in the past. So far more advanced than small molecules, which are effectively chemically synthesized, or biologics, which use proteins. Now we're using whole cells in order to deliver these therapies to the patients, and they come under a different, multitude of names. Advanced therapies, advanced therapy medicinal products, which is the European regulatory term. So I might refer to sometimes as ATMPs. Cell and gene therapies, which is very common in the US, but also globally. I think that's a common term as well. But then there's a range of other names which are often used, which may be slightly more specific. So things like tissue engineered products, regenerative medicines, gene therapy, and gene modified cell therapy. So often if you see this terminology, often it relates back to some sort of cell and gene therapy, or what we'd call an advanced therapy. And what are the role of these advanced therapy medicinal products, or these ATMPs? You can see even I switch between terminology, and this is, as I said, the European regulator's term, the ATMPs. Well, the mechanism of action and the therapeutic aim of ATMPs is to directly introduce genetic material into cells or tissues to correct genetic abnormalities, or to make a beneficial protein which can have a therapeutic effect in the body in vivo. And the way we achieve this is we introduce cells or tissues into the body, be it whole cells or cells on a scaffold, and that will be something that Rana will touch on in the tissue engineering aspect of this lecture, with the aim of having a pharmacological or regenerative effect. So you can start to see here, we're looking at transforming the way we deliver therapies. We're now delivering whole cells. We're trying to correct genetic abnormalities to have some sort of therapeutic effect in vivo. And ideally what we're looking for is a pharmacological, or ideally a regenerative effect. So we're trying to regenerate the body back to good health, so that actually it not only treats the underlying symptoms, but it cures the underlying symptoms. So there's kind of two main therapeutic modalities. We can either use what we call wholesale cell therapies, and there is a blurring between the lines. I'm not going to go into that. CAR T is a bit of a blurring of the lines, which you'll see, but we can either use wholesale cell therapy, so using, for example, stem cells, where we manufacture the cells in a lab and we can deliver them to a patient to have some sort of regenerative effect, or we can try and correct some sort of genetic abnormality, and this is what we would often call gene therapy. And we have both in vivo and ex vivo gene therapy, which I'll discuss on this slide here. So what do we mean by in vivo and ex vivo gene therapy? So with in vivo gene therapy, what we're looking at doing is taking the functional DNA, so taking the gene of interest that we'd like to deliver, putting that into a virus of some sort, so adeno-associated virus is often used, and then delivering that directly to the patient, where the adeno-associated virus would then go to the place where it needs to get to. And that's based on viral tropism, which we won't go into today, or it can be localized delivery, particularly to the back of the eye, and there'll be an example I give later that introduces that. And so we can deliver that, and that would be considered in vivo gene therapy. We also have ex vivo gene therapy, which is where we actually take the cells from the patient. So we take, in this case, let's say T cells or blood. We can take blood from the patient, isolate the T cells. We have the patient cells in the lab. We can transfer, again, predominantly using a virus, we can transfer the DNA or the material into the cells, and then we can deliver the cells back to the patient, which would then be very much a personalized type of gene therapy. But what I want to just give you a quick overview of the kind of the key events within gene therapy development. So gene therapy is not new. It's been around for well over 30 years. So we had the first gene therapy clinical trial treating ADA SCID back in 1990. So, over 30 years ago, we had the first gene therapy clinical trial. And I call this the early clinical phase of kind of gene therapy development. A lot of the research stopped because of the death of a patient, Jesse Gelsinger, who was a patient on a clinical trial. And because of some of the issues around the viruses that were being used, it led to the death of a patient. And it really put a stop on all of the activity within gene therapy, because we realized perhaps we're moving too fast. We hadn't undertaken the necessary long-term safety studies that are required for these types of novel therapies. And we need to be more mindful of that. However, during that period from kind of 1999, so kind of early 2000s, up until about 2010, 11, and 12, there was lots of research being done to try and improve that. And we then had in 2007, the release of the European Union's ATMP regulation for cell and gene therapies. In 2011, we had the first CAR-T clinical trial. And by 2012, we had two major kind of outcomes. The first ever gene therapy approved in the history of the EMA or Western regulators. So Glybera was the first ever gene therapy product approved by the EMA, but also Novartis, who were keen to get access to this CAR-T product, partnered with the University of Pennsylvania to license the CAR-T product that was being developed by the University of Pennsylvania, which we'll come onto in just a second. And this really then kind of from the mid 2010s, kind of 2015 onwards, has seen a rapid transformation and acceleration particularly in the gene therapy space. So having gone from never approving a gene therapy in its history, the US FDA approved 3 gene therapies in the space of 6 months within 2017. The first one being Luxterna, which is a gene therapy for the back of the eye, and I'll show an example of that, as well as Kymriah, which is the Novartis CAR-T product, which you'll see a video on shortly. That was in 2017. In 2020, there were now over 1,000 companies in the advanced therapy sector, and it's gone even further. So in 2021, the US FDA has recently approved Bluebird's CAR-T product, which is, so the Kymriah product treats acute lymphoblastic leukemia, which has a relatively small patient size, but now Bluebird's CAR-T product with Bristol Myers Squibb is being approved for multiple myeloma, which is a far more significant clinical indication moving forward. So things are really gathering pace, and we're starting to see the impact of these therapies. And these are very much what I would say is 21st century healthcare. These are truly personalized therapies where we're taking the cells from a patient, we're processing them in the lab, doing some genetic modifications, and then delivering them back to the patient. So these are patient-specific, personalized, and it's what we like to call vein-to-vein manufacture. So the manufacturing process starts with the patient. So we're collecting their cells. They've obviously gone through a number of treatment rounds before, so their cell quality may be quite low, but we have to try and ensure that we can try and manufacture their cells, genetically engineer them, so we can deliver them back to the patient. And what I like to show very briefly is a video of just how transformative these therapies are. So just to give you some context, this is Emily Whitehead, who at the time, I think she was 6 or 7 years old, was a patient who was suffering from acute lymphoblastic leukemia. So to put that into simple terms, it's a blood cancer. So it's a cancer that affects the B cells. And the normal treatment option is to have a bone marrow transplant. Emily had one and it failed. She was then required to undergo, the next treatment option is then to have multiple rounds of chemotherapy. And as you can tell by the fact that Emily in this image has no hair, she had been through multiple rounds of chemotherapy, but it failed and the cancer kept coming back. And at this point, there was no other treatment option. She was going to die of her disease. But because the colleagues at the University of Pennsylvania were developing this novel CAR-T therapy, she was enrolled on this clinical trial. So for me, I think that is, I mean, I've seen that video now a number of times, and it always reminds me it's why I do what I do, and why, you know, what drives me in terms of my work, and what drives both the teaching activity as well as the research activity, because to have that transformative effect where you can take particularly a pediatric patient, a child who is otherwise going to die of their disease, to be able to provide them with a potential cure. And it is, I mean, CAR-T therapies for a number of patients, not all patients, but for a number of patients, is curative in that it's completely eliminated their cancer. And so this is Emily a few years later, where she had the opportunity to meet the then president of the US, Barack Obama, and just to kind of show you her strength of character and kind of her priorities. She wasn't allowed to tell anyone where she was going because she was meeting the president, and it was for national security. She wasn't allowed to give any details away, so she couldn't even tell her school. So when she met the president asked, "Is there anything I can do for you?" And you can imagine if any of us were put in that situation, we asked for a number of things. She asked, "Could you write me a letter to let the school know that I was with you today, and I wasn't just kind of missing school?" And so that's the letter that Barack Obama wrote. "Please excuse Emily from school. She was with me," signed Barack Obama. So it just kind of shows you the real human element as to why we do what we do. And that is very much a focus for us at UCL. So at UCL, we have an active CAR-T. We're, I think, the largest, certainly within Europe. I think second or third only to University of Pennsylvania for research in CAR-T therapy production. So, some of these articles I'm about to show you highlight the amount of activity that is going on at UCL. And there was a fantastic documentary, which should still be available, I think, either on BBC iPlayer or you can probably get access to it online, called War in the Blood, which is an hour-long documentary showing patients in the UK who have been treated at UCL with the CAR-T therapies, and it shows the patient journey. So it's something I'd highly recommend those of you to watch who are interested in this. But UCL within the advanced therapy or the ATMP space is certainly leading. So this is data based on information from 2017, and I'm sure we're getting up-to-date information now, and it's an even more starker contrast. But UCL was running more clinical trials than every other university in the UK combined. And the other way to kind of frame it is that the UCL was running more clinical trials than the entirety of Italy and Netherlands. So huge amount of clinical activity that is ongoing, but it's necessary that we back that up with the manufacturing capability and activity, which I'll come on to in just a second. I mentioned, so it's not just CAR-T therapies, and which is an ex vivo gene therapy. This is an example of an in vivo gene therapy, so Luxterna, which is used, which is produced by Spark Therapeutics, and is used to treat a rare inherited eye disease, where patients start to lose the ability to see, and it leads to blindness, and it's because they have a faulty gene. And the clinical trials have been hugely successful. This is now a product that is available on the market. And the first patient was treated in the UK, again at UCL Moorfields Eye Hospital and Great Ormond Street on the 17th of February. There was an article of the patient who had restored a significant proportion of their vision as a result of receiving this novel therapy. But the challenge is that these therapies are not cheap. And the focus for us at UCL, particularly within the Department of Biochemical Engineering, with our biochemical engineering and manufacturing mindsets, is how can we reduce the cost of these therapies, and how can we increase global accessibility? So that CAR-T therapy I referred to is over $475,000. Luxterna, again, is well over $500,000. Glybera, one of the first, which is no longer available, but was unfortunately taglined as the most expensive drug in history, over $1 million. So these are not cheap therapies. So the challenge we have is how can we address some of the manufacturing issues to bring down the cost? And this is also a concern for the FDA, where the commissioner of the FDA, Commissioner Gottlieb, highlighted in 2018 that the greater challenge for cell and gene therapies is not necessarily showing clinical promise or scientific potential, but it's a limitation in manufacturing capability for cell and gene therapies. And he highlighted even further that we have to establish scalable manufacturing processes. And so at UCL, a key portion of our research, and so this is a £10 million government-funded manufacturing hub, which is based within our department at UCL, called the Future Targeted Healthcare Manufacturing Hub, where a significant proportion of our research is focused on addressing and developing personalized therapies, in particular the cell and gene therapy. So moving from kind of a one-size-fits-all approach where, we can all buy, let's say, paracetamol or aspirin, from the pharmacy. It's not, there's no aspirin directly made for me. There's no aspirin directly made for any of you. We all buy the same box of aspirin and we take the same medicine. But how can we start to develop targeted medicines? So the medicines that will really take into account my own personal condition, my ethnicity, my age, whatever it might be, to ensure that I can get the most from that particular medicine. And that's what we're starting to move toward. So this is a research hub focused on addressing the manufacturing challenges of developing personalized therapies. You can imagine there's a number of manufacturing issues we have to address. So we lead it at UCL, and we work with a number. So we've got over 45 companies who actually pay to join this hub as well. So we not only have £10 million from the government, we have significant cash contributions from a number of the key companies, be it GSK, be it Fujifilm, AstraZeneca, Allergan, Autolus, a number of other companies who are active within this sector. But I want to finish off my section with a couple of final slides about skills. So it's not just about doing the research and having the manufacturing capability. The other key challenge that we're finding within the sector, and this is an example of gene therapies and the viral vector shortage, is we lack the capability. So there aren't enough manufacturing facilities, although that is now changing. But you can have all the manufacturing facilities in the world if you don't have the right skilled expertise to be able to work in these facilities and to lead these facilities. And that's really what I see as critical to our role, as well as training the next generation of leaders, scientists, engineers, business professionals who can drive this sector forward. And there is an absolute shortage. You can see here from this article in Stat that an employee was offered stocks, sky-high salaries, and a $10,000 watch just to get them to join the company. And startups are desperately competing for top experts in cell and gene therapy. And so you can start to see there is an absolute need for skilled engineers and scientists. And this is highlighted certainly even on the UK front. So the UK Cell and Gene Therapy Catapult, which is a government-funded organization to support cell and gene therapy development, highlighted that 98% of companies are increasing their headcount within the next 5 years, and 83% of those companies have highlighted that they have raised significant concerns that recruitment or retention of skilled individuals will be a key issue for growth. And just taking one of those graphs from that report, you can clearly see that by 2024, so not even 5 years from now, by 2024, we're going to require an increase in jobs there's expected to be an increase of over 3,000 jobs. And a significant proportion of these jobs in green are going to be in bioprocessing, biomanufacturing, bioprocessing, and biochemical engineering. And that's really, for me, what I see, in addition to leading some excellent research, which I won't go into, but I'm more than happy to in our session on Friday, where we introduce the master's program in a bit more detail. Our other key focus is addressing the skills shortage. So with that, I'd like to hand over to Rana, who can give a bit more information about tissue engineering, GenMed, and stem cells.
So hi, everyone. So thank you, Qasim, for the lovely talk. Before starting talking about tissue engineering and stem cells, I just want to mention what Qasim already mentioned, that biochemical engineering is like a new field, like approximately 22 years ago. We started it at UCL, but it is really exciting. And speaking from a biochemist background, so I did my undergraduate as a biochemistry and then cancer research in my master's. So when I first applied to PhD, I like the idea of biochemical engineering on how you can combine the biological side with the engineering side. And the most important thing that Qasim spoke about is the cost of the products. So it's not only about the discovery of a treatment, it's how can you make things affordable and reachable for all people, okay? So I lead on the regenerative medicine minor, which is a minor that you can take if you apply for the within the Faculty of Engineering at UCL. And I teach, most of the topics are regarding regenerative medicine. So I'm gonna start with the definition of regenerative medicine because mostly it's like a new term. So basically, regenerative medicine combine the stem cell therapy with the tissue engineering. And the aim of it is to restore, so to give a patient something to be able to restore the tissue or to replace the tissue or restore the function of the tissue. Already Qasim spoke about CAR-T. But today I'm going to focus on tissue engineering and stem cells. So let's start first with the tissue engineering aspect. The principle is very simple. I always tell the students, you will have to create something. So let's say that a patient is suffering from bone loss because of a disease. So what you have to do is you have to take the cells from the patient, or you can take it from somewhere else. You have to expand them in the lab to have efficient number. And then you put it on kind of a matrix or a scaffold. And this scaffold depends on which kind of tissue engineering approach you're doing. It can be a solid one, like for the bone, or it could be like a soft one, especially when it comes to cartilage replacement or skin tissue engineering. So you combine them together and then you give it back to the patient. But it's not as simple as that. And till now, we're still having a lot of problems when it comes to tissue engineering. That's why we're not progressing that fast. Maybe you can see in the market some of the tissue engineering products, especially like the skin patches. But when it comes to other tissue engineering approach, we have a lot of challenges. Mainly, for example, especially when it comes to more complex structure like the heart. So how can you make sure that you create the heart with a specific structure? You make sure that the blood vessels are going there. You have the valves in the atrial and the ventral. So it is super hard to do, and we have a lot of challenges. But we still have most of our products that are in clinical trials. For example, next slide, please, Qasim. So most of the clinical trials going now are for the tissue engineering replacement for the trachea, small bone fractures, and cartilage. So I'm going to give you a case study for the trachea replacement. This patient suffered from a disease called tuberculosis, and she has her trachea collapsed. So what they did is they took trachea from someone else, or you can take it from animal tissue, and then they removed all of the cells from the other patient, and then they took some of this patient's cells, they put it on the top of the trachea, and they give it back to the patient. And this is one of the most famous first tissue engineering approach, and the patient recovered after that. So I'm just gonna briefly show you some of the example how trachea looks in the lab. So in the first one, you can see that they took from here, this is an example where they took trachea from pigs. So what they did is they have to remove all the cells from the animal source, because when you give it to the patient, so the patient's immune system is not gonna recognize that and attack it. So they do something called decellularization, in which you are removing all of the cells. And what they did in the next set of the pictures, you can see that they put the patient's own cells on the trachea in order to grow it. So this is basically the basic approach on how you do it. For us as engineers, we like to make sure that, for example, when you grow this kind of tissue, especially like if it's bigger than this kind of tissue, how can you make sure that all of the cells are equally distributed within the scaffold of the trachea? How you can make sure that all the cells are taking all the nutrients and oxygen? So instead of growing this in a roller bottle that you can see in this picture, we try to create that or to grow that in a bigger bioreactor in which we can control the environment. Okay. So now I'm going to talk about stem cell. I'm a bit biased when it comes to stem cells, as I did my PhD on stem cells. So first, let's talk about the founders of stem cells. Now, we have different stem cells. So we have pluripotent stem cells and we have adult stem cells. In our body, we can only find adult stem cells. That means these are stem cells capable of generating only specific type in your body, like hematopoietic stem cells that can give rise to blood cells. In the another term is a pluripotent stem cell, which is like kind of a mother cell that can give rise to any cell type in your body, but we don't have it in our body. We can only isolate that from the inner cell mass of the blastocyst in the embryo. So the first founder who has isolated this embryonic stem cells from mouse was Evans. And then back in 1998, Thomson was the first professor or scientist to isolate human embryonic stem cells. And then all of the research has started back in 1998, how you can use embryonic stem cells, let's say to create nerve cells or heart cells and give it to the patient. But because of all of the ethical debate, especially that these cells comes from the embryo, the research has been stopped up till 2006, 2007, where the Nobel Prize winner Yamanaka discovered that by taking some cells, any differentiated cells in your body and any nucleated, it has a nucleus, you can reprogram it to become pluripotent stem cells. By doing this, you don't have any ethical debate, you don't have any immune rejection, and then you can give it to the patient. But the whole concept of creating a reprogrammed pluripotent stem cells, or what we call induced pluripotent stem cells, did not come all of a sudden. So there has been a lot of approaches in developing the idea that you can take any cells in your body, reprogram it to become iPSCs, and then differentiate that. So all of the approaches, for example, when they first cloned the first dolly, they discovered that you can, there are some key regulators within the oocyte that can control the cell fate, up to the discovery of certain master key generators in the cells, and of course, after the isolation of embryonic stem cells and the mouse and the human. Okay, so basically, this is what I explained. Embryonic stem cells can come from the inner cell mass of the blastocyst, and the induced pluripotent cells can be reprogrammed from any nucleated cells. Once we have them, so let's say a patient is suffering from Parkinson's disease and they need dopaminergic neurons. So we take a skin biopsy, we reprogram it to become induced pluripotent stem cells, and then we differentiate into these neurons and we give it back to the patient. Okay, next slide, please. Okay. Now, the first clinical trials that involved these induced pluripotent stem cells happened in Kyoto Center, which is Yamanaka Centers in Kyoto University in Japan. And they give the patient the first iPSC's product for retinal transplantation. So this, the patient suffered from loss in her vision. So what they took her skin cells, they reprogram it, and then they differentiate it into retinal cells, and they give it back to her. And the patient showed some progress when it comes to her vision. So there was, after AMD, and you're gonna see later on that most of these clinical trials are related for AMD diseases, which is macular degeneration diseases. Later on, a few years ago, 2 years ago, they started the first clinical trials when it comes to Parkinson's disease, because as you know, usually the current treatment for Parkinson's disease involved a drug called levodopa that just treat the symptoms rather than treating the disease itself, and the patient is showing some recovery. So in the next slide, you can see the progress when it comes to clinical phases. Now, as tissue engineering, we still have a lot of challenges, and even some challenges regarding the regulation that we follow, and we need to make sure that the product has, for example, if I'm looking for neurons, it's 100% in neurons, because if the final product contain any stem cell, these stem cells, when you give it back to the patient, it can cause cancer. So these are the most recent stem cell products in the market. As you can see, they're still in the clinical trial phases, and most of them, they cover the macular degeneration, and some of them Parkinson and spinal cord injury. Thank you. Qasim, you're muted.
I am muted again. I keep forgetting. Thank you very much for that, Rana. And apologies. I think there were some audio issues, and my slides kept jumping around. So apologies for that.
Sorry to say that in the recording we can iron that out. There are just differences in volume. So sorry it was disconcerting, but we will be able to sort that out. We'll sort it out in post-production, but there'll be a link sent afterwards.
Perfect. Thank you very much, Kim. So perhaps then just to wrap up and kind of give a summary and the future outlook. So I think one of the key things for me is when we look at the therapies moving forward and healthcare interventions, in the 21st century and beyond, from my perspective, they're very much going to be focused on new gene therapies, new stem cell therapies, new regenerative medicines, where we're not just treating the symptoms of a condition, but we're actually developing cures. And so, for example, when you look at diabetes management really involves taking some pills which don't address the issue of the beta islet cells or the pancreatic cells being able to produce insulin. Or we, treat many of the conditions of diabetes, so foot ulcers. And the way we treat with the foot ulcers is we use bandages, and that requires nurses to, replace the bandages regularly to ensure they don't get infected. And it's not great for the patient. It's not great for the healthcare system because it adds additional cost. If we can start to develop therapies that either address and allow us to produce insulin in the body in vivo using synthetic cells or using stem cells, that really is going to be a curative approach, similar to the CAR T therapy that I showed earlier, where what we're doing is we're taking the patient cells, genetically modifying them, and then delivering them back to the patient as part of a truly personalized therapy. Likewise, with the Luxterna gene therapy, you can see that's a transformative therapy. These are patients who have a faulty gene that would otherwise be, living in complete blindness because they would eventually go blind and they wouldn't be able to have their sight restored. But imagine a situation where if we're able to deliver this therapy to the patients that need it, where they can start to see, start to have their sight restored. It may not be perfect, but it'll be certainly better than being completely blind. And that's the aim, that's the goal that we're looking to get to using advanced stem cell and gene therapies. But a significant challenge is how can we address the manufacturing issues? So we've got the opportunity now to really address previous unmet clinical need, and in some cases cure untreatable conditions. And for me, the goal, could we start to think about curing all forms of cancer? I think we've certainly showed for certain forms of cancer, particularly hematological malignancies or blood cancers, we're starting to develop therapies that could provide a potential cure. But could we try and develop cures for solid tumors, which is obviously a major challenge at the moment? And now there's a huge amount of work using cell and gene therapies to think about cures for solid tumors. So from my perspective, the scientific and clinical potential is certainly proven. However, we need to solve the biomanufacturing challenges and make these therapies cost effective. So Rana mentioned and gave the example earlier of the patient who had a windpipe or trachea replacement. So that was Claudia Castillo, and she's still living until this day. And that work was done back in the late 2000s. And the thing about that therapy that she received, that was a clinical trial. It was a one-off. It was a very small scale study. That manufacturing process cost over £7 million. There is no healthcare agency in the world that will approve a therapy for patients that costs £7 million per patient. So although that was fantastic for Claudia Castillo, and she, thankfully is living healthy until today, you can see that just isn't feasible for the wider patient population. And that really is the goal for stem cell and gene therapies. And for me, I think I just want to end on this kind of plea, because for me, I think there is a significant requirement for skilled individuals. And so there's absolutely an opportunity for capable, passionate and willing scientists, engineers and business professionals who want to, transform healthcare moving forward and be a part of the revolution of advanced cell and gene therapies, moving forward. And so on Friday, we have a further session focused on the new master's program, which is currently recruiting in the stem cell and gene therapy space, which I can go into more detail about then on Friday. But even at undergraduate level, there's a significant amount of focus in the cell and gene therapy area. And as Rana mentioned, she leads our regenerative medicine minor. So with that, happy to take any questions, and I can see some questions have come through. So very much keen to to see and answer some of your questions that have come forward. But please you feel free to ask more.
So just whilst Qasim and Rana go through the questions, I mean, I've seen some great ones that have come through. I've tried to answer a couple in text as we've gone through, if it's something that I can answer. Just to just reiterate what Qasim said, there's a webinar on Friday. You can talk to Qasim and Rafiq in detail about the MSc that they run. There's a link to that in the chat, so you can register for that at the moment. Just quickly as well, I'll also be posting a link to a survey at the end. We've had loads of questions. We're not being able to get through all of them, so I'm going to leave to Rana and Qasim to choose which ones they want to answer. Qasim, are there any that you?
Yeah, sure. So I'll start going through. I mean, I'll try and take it in chronological order as they come in through as well. So there's a question here: how do you decide whether to use in vivo or ex vivo gene therapy? What are the advantages and disadvantages? So I think it's a very good question. I think we will use both, continue using both, and they do, and it's based on the clinical application. It's based on the so some factors around the manufacture and delivery aspects as well. So, for example, in vivo gene therapy is predominantly used at the moment for parts of the body which are easy to access. So what I mean by that is, like, for the back of the eye, for example, you can have a surgery where, it doesn't sound pleasant, but it's relatively easy to do, where the ophthalmic surgeon can effectively take a needle, obviously under sedation, and transfer and inject directly into the back of the eye to deliver the gene therapy. And there we can use an in vivo AAV. The challenge with AAV, however, is that obviously if we do it systemically into the blood, it may not go to where it needs to get to all the time, especially if there is multiple, the viral tropism isn't hugely specific. Viral tropism effectively means the ability of a virus to go to a particular part of the body. So there are, viral tropism. So some AAV serotypes prefer going to the liver or the kidney or the brain or whatever it might be. So depending on that is an issue. The other challenge we have with AAV in particular is that it has a relatively small cargo size, in that we can't put a large piece of DNA or RNA into the gene therapy, into the AAV. So it's again limited by the size of the gene that we're inserting as well. So that's something that we have to take into account. Ex vivo gene therapy is technically probably easier because we can isolate the cells from the patient and engineer the cells in vitro and then deliver them back to the patient. But that's not feasible for every condition that we're trying to treat. So it does depend. Certainly moving forward, there'll be a combination of both in vivo and ex vivo gene therapy moving forward. Rana, I saw your hand up. Did you want to add to that?
No, I just, it was by mistake, Qasim.
Okay,
I just want to answer another question, so it should be fine.
Sure. So there's another question here, and then I'll perhaps hand over to Rana on some of the cell and gene therapy ones, on the stem cell ones. What A-level subjects are needed to study biochemical engineering? So the ones that we recommend, if you're looking for the B-Eng or the M-Eng course, would be chemistry, biology, maths, and or physics. I think you need 3 of them. You don't need all 4, but 3 of them is kind of what we normally expect for that, and there'll be more information on our website, and Kim can certainly provide more information on that respect. Rana, there's a question here which I'll direct to you. How are the nucleic cells reprogrammed to become induced pluripotent stem cells?
Okay, so sorry about the voice. I don't know what's going on, but anyway, so how you can reprogram it? So usually we use viruses or non-viral method like episomes, and then we introduce 4 genes, 4 transcriptional factors that usually help to maintain the pluripotency, and usually we call these Yamanaka's factor. So by introducing these kind of gene into the cells will be start to express these transcriptional factor and then start to behave as pluripotent stem cells. There was another question. What are the major concerns regarding the use of induced pluripotent stem cells and how we can improve globally? So, I mean, the main thing is that you are making this personalized, so the immune system is not going to recognize it as foreign and get rid of it. The main concern is the time and the cost of the process, because to take skin cells and then reprogram it to become stem cells, it takes around 1 month. And then to differentiate the cells, it takes another month. And then going through all the regulation and the QC and the QAs, it takes a long time, and then it is costly. So this is basically the main concern regarding the iPSCs. I think, Qasim, there is one about the MSc.
Yes, I think questions related to the MSc, I'm more than happy. I think we'll do that today. I think we can cover that in the session on Friday.
I just want to quickly clarify. We've had a couple of questions about IB subject requirements. All the subject requirements are on the website. Just let you know, it's for A levels, it's a requirement of maths and a science for the BEng and MEng, different requirements for the BSc. So it's really worth checking the website for details on the entry requirements. Again, there'll be details about the IB and what's required and language requirements, far more than we can go into here. But if you want to talk about the degrees that we teach, we're doing an open day on the 14th of June, and there'll be the admissions tutor, Dr. Brenda Parker, there. Come and talk to us, and we'll talk to you about the whole program, careers, any degree you're interested in. Come join us. We'd love to see you there.
Perfect. So I'm conscious of time, but we've got a number of questions I'd like to get through if I can. So I might try and combine a couple of questions. So there's a couple of questions around CRISPR. So there's one specific question around, are viral vectors here to stay forever, or do you see the landscape changing with CRISPR and other types of therapy? So certainly, and there's another question around CRISPR as well. So CRISPR, for those of you that don't know, is a very exciting gene editing technology. So the idea that we can edit the genome using these novel techniques. And CRISPR is not the first of its kind, but it's very effective. There are others. There's ZFNs, which stands for zinc finger nucleases, and there's TALENs as well. So there are other mechanisms that can be used to edit the genome, but CRISPR has a certain level of precision, which gives it a certain advantage over perhaps some of the other techniques. Absolutely, they will be a key part of the therapeutic modality moving forward for gene therapies. In terms of will they replace viral vectors, I don't believe that will be the case. I think there's going to be absolutely the need for viral vectors moving forward. And it's funny because we had an industry panel session last week where I actually posed this very question to industry colleagues, and we all agreed that, moving forward, there'll almost certainly still be a need for viral vectors, perhaps not as much now as we are seeing now. So in 5-10 years, there will be certainly an increase in non-viral techniques. But what we have to recognize is what the virus actually does. So the virus does two jobs in one. It allows us to deliver the material to the cell, and it also allows integration, once it's inside the cell, to integrate the genome into the cell, which is why viruses are very effective. With the non-viral techniques, we need to do both steps together. So we need to have some mechanism of allowing the DNA to enter the cell, or the CRISPR RNPs, the ribonucleic proteins, to enter the cell. So there we might use something like electroporation, which is kind of giving the cell an electric shock, which then what that does is, quite interestingly, is it creates pores in the membrane, and then we can kind of push in our CRISPR ribonucleic proteins, and then that CRISPR RNPs will then deliver the gene of interest, or do the gene editing internally within the cell genome. So we need to increase the efficiency of those methods. So those methods, although very promising and eliminates the need for viruses, which links them to another question, which I'll come on to, eliminates the need for viruses. At the moment, they're just not efficient enough, and that will change over time as we move forward, which is one of the key areas for manufacturing optimization. That leads on to another question, which I wanted to answer, which was, why is the cost for these therapies so high? Or this specific question was, which part of manufacturing warrants such a hike in cell and gene therapy prices? A significant proportion of the cost is the viral vector. So the viral vector can range between, I would say, 50 to 100,000 pounds or dollars for just the viral vector itself. So eliminating the need for viral vectors will have a significant impact in the cost of goods. But there's also other aspects. I mean, those prices that you're seeing at the moment, $350,000, $450,000, a lot of that is potentially on the part of the therapeutic company trying to recoup development costs. So, they've been spending millions and millions over the last 10 years developing these therapies. What they're doing in some ways is trying to recoup their investment costs as part of the therapy. But also, even in terms of each individual manufacturing aspect of the process, you imagine a personalized therapy we have to get the hospital to isolate the cells, we have the patient to schedule, we have to then transfer and transport those cells, we have to maintain them under certain conditions, we have to grow them. Significant proportion of the cost is cell expansion, which is the area that I'm particularly interested in and do lots of research on. How can we bring the cost of expansion down, reduce the time, we have to freeze them, and so on and so forth. So all of those have additional costs. And there was another really good question, which I liked actually, is what would the side effects of the treatment be if they weren't personalized? Why is personalized treatment a goal? I think that's a very, very good question. And the question that I often ask is, personalized therapies are very expensive. Why is that the end goal? Are we getting a personalized treatment? For many therapies, we don't need them to be personalized. We don't need the aspirin that we take or the paracetamol that we take to be personalized. But for certain therapies, we do. So with the T-cell CAR T therapy I showed earlier, T cells, if you transfer them from one patient to another, will cause an immune reaction. And one of two things will happen. You'll either have graft versus host disease, which where you'll have immune reactions with the body either destroying the treatment, the immune system, or potentially the immune cells that are being delivered to the patient from another patient having a harmful impact on the recipient, and therefore you'll have lots of side effects and immune reactions, like similar to what you have with transplantation and then having to be on immunosuppressants for the rest of your life. So there's a number of concerns around immune reactions from transferring these cells from one patient to another. Some cell types, and this is an area that I'm particularly interested in, such as NK cells, natural killer cells, can avoid the immune system and don't necessarily have the same immune response. So therefore, potentially using that as a therapy would be ideal. I'm conscious of time, and there are a number of questions, some really good questions, I should say. Should we be looking at other viral vectors? How do biochemical engineers choose which viral vector would work best, and so on and so forth, the role of epigenetics and stem cell therapies. There's some fantastic questions here, so I really would like to thank you all. Unfortunately, I think we have run out of time, and I don't want to kind of keep you beyond the stated time. If you have any questions about the programs, whether it's the undergraduate or the postgraduate programs, please do reach out to Kim and find out more information on our website, or feel free to contact myself. As I said, we do have a separate webinar on Friday focused on the master's program in stem cell and gene therapy, manufacture and commercialization. So you're more than welcome to join that as well. And maybe you can address some of these questions there as well. So with that, I would like to thank you all for your participation today. I hope you found this useful. Do feel free to reach out if you have any queries, questions, particularly about the programs and so on as well. And we look forward to keeping in touch moving forward. Thank you all very much.
Thank you very much, Qasim. Thank you very much, Rana. Thank you very much, everyone, for joining. The links are in the chat at the moment. I've posted a survey questionnaire as well. I thank you for your kind comments. Very much appreciated. And I hope you have a great afternoon. And I really do look forward to meeting you in person. We all look forward to meeting in person, hopefully at some point in the future. All the best. Thank you. Goodbye.
Fantastic. Thank you very much, everyone.
Thank you, everyone.